Hi everyone, it's my great pleasure to talk about high quality plasmid DNA manufacturing strategy for advanced therapeutics. Today's presentation, I have divided the talk into 4 parts. The first part, I will talk about the plasmid DNA market in advanced therapeutics. The second part, I will talk about how plasmid works. The third part, I will talking about the manufacturing strategies of plasmid DNA. Last but not least, I will talk about the GenScript ProBio one-stop solution for plasmid DNA. So, the global market for advanced therapeutic products, we have summarized those commercialized products in this table. As you can see, starting from 2005 to 2016, over 10 years, only 3 therapeutics has been approved and commercialized. But from 2017 to 2021, you will see 9 therapeutic drugs has been approved, and that shows that for the past 4 or 5 years, the advanced therapeutic market is really growing at a very fast pace. From the market perspective, you will see that the plasmid manufacturing market and the viral vector manufacturing market steadily grows over the years. As you can see in this pie chart, 2019 we have the sales, the market is only around $1 billion US, and for the prediction in 2030, this market will increase to almost $6 billion. That means the market for plasmid is really big. The second part, I will talk about how plasmid works. So plasmid has been used widely in gene and cell therapies. Here we categorize the application into 4 different applications, such as cell therapy, gene therapy, mRNA products, and DNA vaccines. For the cell therapy products, either you can use plasmid DNA to produce lentiviruses and use that for CAR T cell therapy, infuse the cells into patients, or you can use stem cells, induce pluripotent stem cells, and engineer those cells with plasmid and get the infector cells infused into patients. For the gene therapy, either you can use naked DNA as a non-viral vector directly inject to the patients as a gene therapy product, or you can use plasmid DNA to produce, for example, adeno-associated viruses, and use this as a viral vector to deliver a gene to the patients to treat the diseases. For mRNA products, you can use plasmid DNA as template to produce mRNA, and this mRNA can be used to treat cancer and to prevent infectious diseases such as mRNA vaccine. Recently, most people know about for the COVID-19. DNA vaccine works similar way, but instead of using mRNA, you can use DNA, and I will talk about how this works in the later slides. So here is the slides talking about how we use plasmid to produce lentivirus and adeno-associated virus. For lentivirus and adeno-associated virus, in short AAV, in industry, most people use transit transfection. For lentiviruses, researchers are using third-generation plasmid, which contains 4 plasmid, as shown here, including transfer vector, packaging vectors, envelope vectors, and you use those vectors to co-transfect hack 293 T cells and use cells as factory to produce lentiviral particles. Those particles in size is around 120 nanometers, and it carried the gene of interest. Either you can use it for CAR T cell therapy, or you can use for other cell therapies or gene therapies. For AAV, instead of using 4 plasmids, AAV typically using 3 plasmids, including 1 transfer vector and 2 packaging vectors. And you can use these 3 vectors at the same time to transfect the 293 cells and use this cell to produce AAV particles. Those AAV particles are much smaller compared to lentiviruses. AAV particles are only around 20 nanometers with capsid protein cover it. And for the mRNA products, here I want to show mRNA vaccine. As everybody knows, that recently mRNA vaccines has been approved by FDA and EMA to prevent COVID-19 pandemic spreading. So mRNA has been proved to be able to express protein when injected to the animals in the early 1990s. It's been a long journey, and finally mRNA can be used as a vaccine because mRNA not stable, easily degraded by DNase. So over the years, researchers have been able to modify the mRNA and make it more stable. If you look at the IVT mRNA structure, the in vitro transcript mRNA typically contains 5 parts, including the 5' parts. You have a cap, and the 3' ends you have poly A tails, and in the middle you have so-called open reading frame ORF. This part is coded the protein of interest for vaccination or therapeutic purpose. Typically for the COVID-19, you can code in this part you can code the spike protein and use it. So how mRNA works? There are 2 types of mRNA. Either it's non-replicating mRNA or self-amplifying mRNA. These RNA, since they are not so stable, typically for delivery purpose, you encapsulate these mRNA molecules into a delivery system. Most commonly, the lipid nanoparticle are being used. This lipid nanoparticle carrying mRNA will be engulfed by the cells simply by endocytosis. Once the particle enter in the cells, mRNA will be released, and according to the central dogma, it will be expressed as protein of interest. This protein later being secreted or released from the cells will being recognized by T cells or B cells and generating immune responses. So this is the way how mRNA vaccine works. Similarly, for DNA vaccine, in this case, instead of using mRNA, you use DNA as vaccines. DNA has to go through 2 barriers instead of 1. mRNA only need to go through the cell membrane, but the DNA have to go through both cell membrane and the nuclear membranes, and then being translated to mRNA, and mRNA will be expressed as protein of interest, and this protein will be released from cells and being recognized by T cells or B cells to generate immune response. So this is how DNA vaccine works. After I talking about the principle how this plasmid has been used for gene and cell therapy products, I want to switch gear to talking about how we are going to produce those plasmid DNA. Typically, the plasmid manufacturing process is showing here. It starts from fermentation in a bioreactor, and then you can harvest the cells, lyse the cells, go through clarification, concentration, and then go through purification steps using chromatography, and then concentrate by UFTF, and then finally formulate the plasmid DNA products. And the sample, of course, will be tested by quality control department and released for later use. And the whole process can be divided into upstream and downstream. Upstream typically including the part for fermentation. You can optimize many factors as shown here, including the culture media, including the optimizing the feeding strategy, pH, all these parameters. You will be able to increase the yield of plasmid. Here, just give you the example. As you can see, when you vary the pH from value A to value B, you are able to increase the plasmid yield significantly. And another factor, you can change different host cell based on the vector, right? If you use the same vector but changing different host cells, you'll be able to get the different yield. And then from here, you can choose the best one to produce plasmid DNA, because this one give you higher yield. After optimization, you will see that our upstream fermentation process has been optimized, and we can easily get high density fermentation and reach a product easily have more than 500 milligram per liter or even higher. And the whole process, as you can see from those graphs, they are highly stable and have very low batch-to-batch variation. So for the downstream, the major part is purification steps, typically using chromatography. For separating product plasmid DNA from the impurity, you need to understand the structure and the chemical characterization of plasmid DNA. First, this plasmid DNA, they are circular and they are double-stranded, and the molecular weight is about 1 million to 10 million Daltons, and it has other physical and chemical characteristics, and you can use that to separate other impurities. I'll give you an example. For example, the isoform of plasmid DNA, they have linear plasmid DNA, open circle plasmid DNA, or supercoiled plasmid DNA, which here the supercoiled plasmid DNA is thought to have a therapeutic effects, and you can use affinity column or hydrophobic interaction chromatography to separate them. Another important impurity you need to separate is RNA. So based on characters such as charge, you can use anionic exchange chromatography, or you can use size simply because mRNA, RNA and plasmid DNA have different size. You can use size exclusive chromatography to separate these two molecules. Here I want to show the case how we improve our purification process. As you can see in this graph, we can get a pretty high recovery rate. Typically, based on the project, we can get a recovery rate between 20 to 50%. And here it shows the final yield. We can also achieve pretty high yield. Let's say for a 10-liter batch, we can achieve more than 700 milligram easily. And in terms of impurity, we can have a very high pure product. The impurities such as whole cell DNA, we can achieve the content for HCD below 0.2%. For the whole cell protein, we can achieve in the final product is less than 0.0003%, so which is very little HCP left in the product. And for therapeutic part, the supercoin, the plasmid product, we can get the supercoin content up to 97%. For the plasmid DNA, we also have very strict release criteria as shown in this table. I don't want to go to the detail, go through the line one by one, but I assure you we can get much more stringent release criteria compared to what FDA or EMA required for. Here on shows one case study, and the case study we choose, since many of you are interested, is use plasmid DNA as template for mRNA vaccine production. Here, the first thing we want to show that we can optimize our process to achieve much better yield. You can see after optimization, we can achieve almost five times more product after the optimization. And for the batch-to-batch variation, here I show two projects we have done. You will see that the plasmid yield is almost same. One of the important characteristics is mRNA containing poly A tails. As I mentioned previously, having these poly A tails enable to stabilize the mRNA molecules. And for our product, you can see we are able to make plasmid with poly A tail length varying from 80 nucleotides up to 150 nucleotides. And we also have excellent stability. As you can see that we have done 50-liter manufacturing without any problem. And the loss rate for poly A tail is less than 5%. Another case study I want to show is the plasmid for lentivirus packaging. So we have ready-to-use lenti-helper plasmid, as shown here. This plasmid has been registered with CBER FDA, and they are immediately available to support your IND application or early phase clinical trials. And for the packaging efficiency, we have used those ready-to-use lenti-helper plasmid in both suspension and adherent culture to package lentiviral vectors. As you can see, we can easily achieve high titers of lentiviruses, easily above 10 to the power 7 TU per mil for both adhering and the suspension culture. And especially for the GFP genes, we are able to produce titer above 10 to the power 8 TU per mil, which is really high. So this once again show that our ready-to-use lenti-helper plasmid, they are very efficient in helping the lentivirus production, and they are ready to use supporting IND application. So last but not least, I want to talk about the GenScript ProBio. For those who are not familiar with GenScript, I want to spend a minute to talking about our GenScript company. The GenScript group has 4 arms, each arm specialized in a different field. We have life science CRO, which produce like primers, genes for research worldwide to many universities and institutes and companies. And we also have one arm called the Best Design, and this arm we are producing synthetic biology products, especially enzymes, proteins, and for the Legend Biotech, this arm is focusing on the cell therapy, especially CAR-T technology to bring a miracle to life. So we are positioned in GenScript ProBio. Our company is Biologics CDMO. Our slogan is innovative technology drive sustainable growth. So what is GenScript ProBio? So our vision is to your most reliable and sustainable biologics partner. Our mission for GenScript ProBio is dedicated to proactively providing end-to-end service from discovery to commercialization with professional solutions and efficient processes to accelerate drug development for customers. Through the past 2 years, we have a very solid track record, and we have global partnership with big biotech companies. So far, we have done about 60 projects for plasmid and virus CMC and clinical GMP projects. 7 projects already got IND approval, either from Chinese or American regulatory agency. And we also have 3 mRNA projects which achieved IND approval, and we have over 10 mRNA customers who are producing plasmid CMC projects. We are partner with big pharma and the leading biotech companies, including Korean companies and the Chinese companies. We also provide the plasmid for 3 leading vaccine companies. And here showcase, for example, we partner with X-Life Science to make TCR-T products for cell therapy. And we partner with STEMI RNA and Upstream mRNA to produce, help our partner to produce mRNA vaccines against COVID-19. So this is our solid track record. For the plasmid, actually, FDA, the U.S. Food and Drug Administration, EMA is the European counterpart, NMPA is the Chinese counterpart. Based on the different application, the requirement for plasmid very different. For example, for CAR-T or TCR-T cell therapy, typically you need the plasmid for the lentiviral packaging, and this you need GMP principle, and the plasmid is so-called intermediate. For the other cell therapy, like we use induced pluripotent stem cells, this you need the plasmid to induce stem cells and to be certain program them to certain special cell lines. And this time, plasmid is used as a raw material. For gene therapy, first one for therapeutic gene and like AAV, plasmid, transfer plasmid is considered intermediate product. And for CRISPR-Cas9, we use plasmid as template for Cas9. This time, plasmid is considered as a raw material. And for cell modification, plasmid serves as a non-viral delivery vector. It's considered as a drug substance. And in this case, you need cGMP. It also means current GMP. For mRNA product, plasmid DNA is used as a template for produce mRNA. In this case, plasmid is considered as a raw material. Of course, it's under GMP principle. For DNA vaccine, plasmid is used as a drug. Its definition is as a drug product, and of course, it's under cGMP conditions. So, as a GenScript ProBio, we provide you one-stop solution. We have 3 different grades of plasmid, so-called Pro grade, GM Pro grade, and cGMP grade. For each grade of plasmid, you can use for different phases accordingly. So for preclinical and R&D stage, you can use Pro grade plasmid. And for the early clinical phase R&D filing, you can use GM Pro grade plasmid. For late phase clinical trials and BLA and finally commercial product, you can use cGMP grade plasmid. And accordingly, last slide, so I talk about raw materials or intermediate for them. Preclinical, you can use Pro grade. CMC stage or early clinical phase, you can use GM Pro grade. Late clinical phase or commercialization, you can use cGMP grade. For the drug substance and drug products, each stage you can use preclinical, you can use Pro grade. For the CMC and clinical trial commercialization, or you can use cGMP grade plasmid. So this table will help you understand what type of plasmid you need, and as GenScript ProBio, we can provide you those products accordingly, can save you money and time. And the benefits to use plasmid is we call it balance the cost and compliance. For the plasmid platform, we have a very robust platform. As I showed previously, it has achieved high plasmid yield, high recovery, and very low batch-to-batch variation. We have done high-density fermentation. We can achieve high yield between 600 to 800 milligram per liter. We can achieve easily very high content of superchloride plasmid, over 90%. And this system is so-called animal-free and antibiotic-free. And for the plasmid, we have done, we are doing many projects with our plasmid. We produce our plasmid for CAR-T and TCR-T cell therapy, and we have already achieved the IND approval, and we also are doing clinical manufacturing. For CRISPR editing cell therapy, our plasmid are already achieved IND approval. For the gene therapy, we already we are already manufacturing our plasmid for clinical applications, clinical use. For mRNA vaccines, our plasmid already achieved IND approval and reached clinical stage manufacturing for clinical purpose. For virus vaccines, we already reached the stage to produce for clinical manufacturing. And for DNA vaccine, we have a CMC project ongoing. For RLC DNA, we have CMC project ongoing, and a lot more coming up with different applications. Besides our plasmid platform, we also have ready-to-use whole strains and hyperplasmid. For the strains, our strain have clear history, and we can sublicense those cell banks to you without any problem. We have the right. And for the lentiviral helper plasmid, as I mentioned before, those has been registered at FDA CPR. They have clear history and free IP, and those plasmid are royalty-free and maintenance-free, so it can support you for IND application. Those are so-called ready-to-use. With that, I will thank you for your attention. I'll be happy to take any questions.